Speaker mute.
Good morning to one another. Welcome to the international webinar on genes, nutrition and health, organized by Department of Botany and supported by Internal Quality Assurance Cell AQAC and College with Potential for Excellence CPE. With blessings from Almighty, we will begin our webinar. We have two sessions from two young researchers. Our first keynote speaker is Dr. Tapas Pradhan from National Institute of Environmental and Health Research, NIH, North Carolina, USA, and our esteemed resource person. And our second resource person, chief guest, is Dr. Aneesh Kumar Asokan, Division of Endocrinology, Mayo Clinic, Rochester, USA. For welcome address, I would like to invite Dr. Vimala Jose, Head, Department of Botany, St. Thomas College, Thrissur. Over to you, ma'am.
Good morning, everyone. We are here for the international webinar on genes, nutrition, and health. The World Health Organization defines health as a state of complete physical, mental, and social well-being. Nutrition is the most important factor in maintaining and keeping good health. Above all, genes are very much related to nutrition, as the nutrients we consume can impact gene expression and the way our genes are regulated. This webinar, Genes, Nutrition and Health, is being presided over by Dr. K.L. Joy, Principal, St. Thomas College, Thrissur. On behalf of the Department of Botany, I welcome you, sir. Let me welcome our first keynote speaker, Dr. Tapas Pradhan from the National Institute of Environment and Health, USA, who will be talking about cancer, an act of genes and environment. Bearing in mind February 4th as the World Cancer Day, the Department of Botany, St. Thomas College, decided to fix one of our topic as cancer for this webinar. As we know that cancer has remained as a major foe in human health and also as a leading cause of mortality associated with systemic diseases. I welcome you, Dr. Tapas, and very pleased for considering our invitation. I'm happy to welcome our next chief guest and keynote speaker, Dr. Aneesh Kumar Asokan from the Mayo Clinic, USA, a young, dedicated researcher who is very much passionate about science. He will be talking about vaccination and its importance in public health. Now that effective vaccines for COVID-19 have been developed and are being distributed to the public, it's very important to understand the benefits of vaccines and how and why they are greatly outweigh mild potential side effects or inconvenience that may be associated with. Dr. Aneesh, we are privileged to have you with us on the ruling topic, vaccination. A warm welcome to you, sir.
Oh, thank you, ma'am. Yeah.
Also, I welcome our beloved principal, Vice Principal, Reverend Dr. Martin Columbrett, our Vice Principal, Dr. Joby K., and Dr. Sister Alfonsa Matthew, the Dean of Science, Dr. Brito Joseph, the IQAC Coordinator, Reverend Dr. Anil George K., and all the faculties and students from St. Thomas College, as well as from other colleges for this webinar. Thank you.
Hello, good morning to all. Respected resource person, Dr. Tapas Pradhan, Dr. Aneesh Kumar, Asokan, Vice Principal, Dr. Sister Alfonsa Matthew, Dean of Science, Dr. Brito Joseph, IQAC Coordinator, Dr. Father Anil George, Dr. Vimala Jose, Head of the Department of Botany, Dr. Surekha Y. Pawar, faculty members, non-teaching staff, research scholars, and dear students. St. Thomas College Thrissur has been keeping abreast with all knowledge in multifaceted nature, keeping a vigilant mind of accepting innovations. Today, the Department of Botany of our college has come up with an international webinar on genes, nutrition, and health. Dr. Tapas Pradhan, a postdoctoral fellow from North Carolina, USA, guides us along the topic cancer, an act of genes and environment. Cancer can be due to some changes occurring to certain genes and the change in the normal function of cells. Some other cases may result from environmental exposure, namely the exposure to radiation or smoke. Chemists and researchers have presented a list of such toxic compounds. Dr. Aneesh Kumar, Mayo Clinic, Rochester, USA, will lead us along the line of vaccination, the cornerstone of public health. As we know, vaccines play an important role as an efficient tool to prevent infectious diseases. Virus-related cancer can even be restricted with the help of vaccination. Now, the pandemic COVID-19 has made us look earnestly to successful vaccination. It gives me great pleasure to congratulate Dr. Vimala Jose, the head of the department, and the faculty on the conduct of this webinar. Let me express a sincere gratitude to Dr. Pradhan and Dr. Aneesh Kumar for their guidance for our intellectual exercise. Hope our students benefit very much from these academic sessions. Wishing everyone all the best. Thank you very much.
For felicitating, it is my pleasure to invite Dr. Sister Alfonsa Matthew, Vice Principal, St. Thomas College, Thrissur.
Respected Principal, Dr. Joy KL, Head of the Department, Botany, Dr. Vimala Jose, Dean of Science, Dr. Brito Joseph, the resource person for the day, Dr. Tapas Pradhan, National Institute of Environmental and Health Science, North Carolina, USA, Dr. Aneesh Kumar Asokan, Mayo Clinic, Rochester, USA, and other faculty members, and my dear students. A warm greetings to all. It's my pleasant privilege to speak a few words on this international webinar on genes, nutrition, and health. As Gandhiji once said, the world has enough for everyone's needs, but not everyone's greed. When 90% of resources are used by an elite 10%, an imbalance is bound to happen. Famous Canadian historian Early Grey says, most of the 7 billion people in this world suffer malnutrition. Half do not have enough to eat, and the rest of us eat too much. The modern must have, or we can say the pragmatic economy, is eating to the planet's resources like never before. I think this webinar on genes, nutrition, and health is a step in the right direction to make us aware of the grave situation of the world and our responsibility in this regard. I take this opportunity to congratulate and appreciate the efforts taken by the Department of Botany for having organized this event. And I hope this event yields the intended results. And I wish all the success to this event. Thank you.
Thank you, Dr. Sister Alfonsa. Moving forward, I would like to invite Dr. Brito Joseph, Dean of Sciences, St. Thomas College, for the felicitation speech.
A warm good morning to all. Respected Principal, Dr. K.L. Joy sir, distinguished guests, Dr. Tapas Pradhan, Dr. Aneesh Kumar Asokan, who is delivering the keynote address, Vice Principals, Head of the Department, Dr. Vimala, IQAC Coordinator, Father Anil Kongoth, the Coordinator of the program, Dr. Anto, and distinguished guests, my dear colleagues and students. First of all, I would like to congratulate the Research and Postgraduate Department of Botany for coming forward to organize an international webinar on genes, nutrition, and health. Recent studies indicate that there is an interaction of relationships between nutrition and human genome. Nutrition may affect the health condition of individuals, making them susceptible to diseases like cancer, obesity, diabetes, heart disease, and mental illness. In this international webinar, we have two eminent speakers from USA, Dr. Tapas Pradhan from National Institute of Health Science and Dr. Asokan from Endocrinology Division. I hope the resource persons will enlighten us with novel ideas on recent advancements attained in this field. This will be very much helpful for our students to derive a clear perspective to lead a healthy life in future. Once again, I congratulate the Botany Department and wish you all success for this international webinar. Thank you.
Thank you very much, sir. Now let's start the session. Our first keynote speaker is Dr. Tapas Pradhan. Before his presentation, let me introduce him. Dr. Tapas did his bachelor's and master's degree in biotechnology from Utkal University and University of Calicut. He did his PhD from Rajiv Gandhi Center for Biotechnology, Trivandrum, on role of cancer stem cells in colorectal cancer progression and exploring their novel regulators. He has received Young Investigator Award at ISOBM meeting, Hamburg, Germany in the year 2018 and also selected for RGCB Merit Award. He has trained more than 40 MSc and BSc students for their thesis in basic cancer cell biology. He will be talking about cancer, an act of genes and environment. In his talk, he will be discussing on updates on basic cancer biology and role of environmental factors on cancer onset and progression. The presentation will also have a brief part on how different branches of basic biology is critical pillar in the development of future drug discoveries. I invite Dr. Tapas Pradhan to handle the session. Over to you, Dr. Tapas.
Yeah, hi. Hope I am audible to everyone. Yeah. Good morning, everyone. I would also like to welcome you all to this beautiful seminar series. And thanks, Dr. Surekha, for such kind introduction. And I would like to thank Dr. Vimala for hosting this event, Dr. Joy, principal of the St. Thomas College, for hosting this webinar series. And I would not consume much more time, and I would straight go to the presentations. And so let me share my slides. Is it visible? Is it visible? Can I?
It is not visible.
Is it visible now?
No.
Nope.
You have to share your
Yeah, I
screen
have shared it.
and just minimize your live screen actually,
Okay.
and
Okay.
then maximize it, your PPT screen.
Mhm. So yeah, I have minimized the live screen, and is it good now?
It is not visible actually.
No.
1 minute. Sharing that in minute.
So I have gone to share and
So,
share screen.
so you can press on the share button,
Yeah, I pressed it and I
and
shared. And
you can choose the share screen option.
yeah, your entire screen. I have.
Yeah.
Shall I do that?
Yes, yes, please go ahead.
Yeah. So is it okay now?
Uh, 1 minute. 1 minute. Oh, okay, I will do that, sir. 1 minute.
With Tapas, it will start. Okay, so you just continue your presentation.
Is it whether my slides are visible?
No, it will be visible soon. You just have to start. You can start. You can give the introduction. Sorry, thank you.
Is it? Can I start? Is it visible now?
No, it's not visible.
Oh,
You have maximized it, right?
yeah, yeah. I have maximized. I have kept in slideshow. Okay. Wait a minute. I'm sharing my PPT. Is it visible now?
It is not. It will be soon visible.
Hello.
Hello, Dr. Tapas. Hello. Hello, Dr. Tapas.
Unmuted.
Is it okay? Yeah, so I hope everybody can see my screen, right? Okay, so I'll straight go to my slides. Thanks again for inviting me to talk on this particular topic. Oh God, I cannot do it right. Oh. Hello, am I audible?
Hello.
Everybody can see my screen. Actually, I don't know. Can you hear me? Can you see my screen?
Yes, we can hear you actually.
We can. What about screen? Can you see my? I have shared it. Now it is shared. Can you see my PPT?
You can move the. Yes, you can. We can see your PPT actually.
So when I'm talking, can you hear it?
So you have to say next. Then only I can control it. Yes, you can control.
Yeah, I'm. Now can you see? See, now I am sharing. Can you see? Is it?
Yes, yes.
Now, next slide came, right?
Yes, you have to tell me, then only it will happen. Next, you have to say next, then I will do it. Okay.
I cannot do it here. Now you cannot see me.
No.
Oh, okay.
I can see everything, but this is the thing that I have to do it.
Oh, what about now?
Control is in my.
Oh, what about now? I am sharing. It is not possible. It is showing I am sharing the screen now.
Now it is possible that he can share it. So you share it then.
I shared it. Can you see me? That is what I am telling. I have already shared it. Can you guys see?
So I will stop.
Hello, sir.
Yes.
Yeah, I can.
You
hear.
can control. You can control.
Yeah, no, everybody can see, right? Okay.
Yes.
Yeah. Okay. Thank you, sir. Thanks a lot. Okay. So I'm audible, right? And my PPTC is visible, right? So hello. Hello. Yeah. So topic of my presentation is cancer, an act of genes and environment. And it has been put out that what is cancer, and this audience, I think, doesn't need a broad introduction on this disease. And so straight I would go to how these two compartments, genes and environment, interact, and how together they regulate this particular disease, its onset, its progression in human. So before going to that, we would just discuss a little bit about its history, how old this disease is. So mostly we will be hearing that cancer is something which modernization has brought, the modern culture has brought, the modern lifestyle has brought. So is this a disease of modern time? That's the question. So if we look back, and researchers have recently discovered that the first description of cancer has been found in which dates back 3000 BC. And as you can see, it is in Egyptian scriptures, where they have mentioned about how the ulcers of breast, and those were removed by their one technique called fire drill. And the writing says that the disease is, there is no treatment for this disease, right? And so we know that this disease is not which the modern time has brought, or the modern, it was there for a long time. And obviously we now know that disease incidence has increased, the incidence rate has increased, because maybe because the reportings are more, and people are more aware about it. So let's go to the recent statistics of this disease, which this is from the WHO 2020. And as you can see, the global incidence rate, if we see here, the dark blue countries, where the dark blue stains, where this shows the highest incidence rate of overall cancer. Whereas if we come to our country, India, but it is still coming under a low incidence rate country. That means the incidence rate, and that is partially because we do not have a robust diagnosis system all over our country, and which is partially responsible for less reporting, and thus we think that the incidence rate is less, but that is not the truth. That is not the truth. And we can see on the left panel, we can see the number of different cancer categories. The breast cancer, cervical cancer, and head and neck cancer, the lip, oral cavity cancer is leading in our country. And the lip and oral cavity cancer, it could be because of the high tobacco consumption, and which is there in India. And if you come to inside India, the different district which recently has been published, we can see that there are, two districts from Kerala itself, which are coming in top 10 incidents. First one is Thiruvananthapuram. In case of male, it is coming on top, and Kollam. So here in female case also, Thiruvananthapuram is standing along in top 10 highest incidence rate district in our country, which all is a major concern, because the cases are keep on increasing, and we have to be really careful. We have to know, we have to be aware about this disease. So let's come to the definition of this very disease. What is cancer? So cancer is a group of disease which originates from a single cell due to malfunction of genes associated with various bioprocess of the cells, which enable the cell to divide and grow uncontrollably and metastasize to other part of the body. So, which I have highlighted here, red highlighted, are the three important terms in this definition. It divides uncontrollably, it grows, and it can metastasize. I'll be coming to it in a minute, each one by one. So let's first define, when we see cancer, and sometimes we confuse it, what is tumor, what is cancer. So here on the left side, we can see a plant is having a bulk mass, we can see. And as the majority of the audience today in the presentation belong from botany and botanical background, so this figure doesn't need much of introduction or description, that plant do have tumor, and but that is because of the biological agents which infect and the cell grow. But is this cancer? No, plants do not have cancer, because as I defined in the previous day, to call it a cancer, a cell must metastasize. Metastasize means it should move out of its basal lamina or cell. It should come out of its position. It should travel to other parts of the system and again grow in different part of the body. Then only we can call it a cancer. As you can see here, here is a clear defined architect of the lumen and normal ductal cells, where once the benign tumor, you can see a growth of this epithelial cell. However, these cells are not breaching yet the basal lamina. That means they are still contained. Once these cells become malignant and they go to the, that tumor become malignant, they tend to break this basal lamina and move out of their tissue system, and then that is called metastasis. That they travel, and then they can go to different part of the body, and there they can again grow and form a tumor. So this is what happens in most of the animals and including human, and the cause of this are various. How the cancer comes: different environmental factor, which are ultraviolet radiation, different carcinogens which we exposed from different pollutants or say from environment, and viruses, bacteria, which are biological agent. We'll be talking it later in this presentation. And two obvious things which cannot do anything about it is faulty replication, which happens inside our body in a daily basis, and aging. These two are the major factor for cancer also, because the more you live, there is a more chance that you might get cancer. So what are the causes? What causes cancer? So to talk broadly, we have three important compartments where the genes are playing a role. The DNA, which is the blueprint of life, where all coding information is there, what proteins to be formed, what genes to be synthesized, and then epigenetic, which is a very interesting field where the modification in the DNA gives rise and they regulate the gene expressions in particular cell. The methylation, as histone, histone deacetylation, acetylation, all these post-translation, post DNA modification also plays very important role in differential gene regulation, and further leading to altered signaling cues. And this any of the compartment get altered by environmental factor or from UV or any biological agent, then there is a chance that cell will become malignant. Coming to environment, this environment cannot directly cause cancer without going through this compartment. Like it has to alter DNA or epigenetic mechanism. It has to change the epigenome, or it has to change the signaling cues, which fuels the cells, and then only it can cause cancer. So there are all the carcinogens which we'll be talking. They change in any of these three compartments, and then they lead to carcinogenesis. So first, we'll be talking about genes and cancer, how they are important. So genes doesn't need any definition, because genes are the trans element which DNA, from DNA we form RNA to protein, and we all know the central dogma. And so the genes are the regulatory factor for all function, all phenotypes which a cell shows. So basically they maintain all the cellular homeostasis. They regulate everything. To put into simple form, I have here drawn a simple depiction of how the control mechanism works in cell, and by explaining this traffic, a very organized traffic here, and that leads to unorganized traffic. As you can see, sometimes that happens, not always. And how it happens: when the traffic controller fails, or we have the signals, or we have certain rules in traffic, the lanes which people have to obey, that vehicles has to obey certain rule. If they break that rule, they do not obey the traffic signals, or the traffic signal fails, this chaos can happen, and we can see a chaos or unorganized traffic. To put similarly, a healthy tissue also has a very defined architecture, just like this organized traffic. You can see a defined, this is the image from a colon histology, and you can see there are defined structures there. They are very proper, boundaries clear, and there is a proper morphology. However, when it becomes cancer, it's a chaos again. We cannot see, this architecture is totally different. And that also happens because of the genes which controls everything. These are the control of our body. These are control of our cells. If they get altered, they alter the signaling pathways, which maintains these healthy tissues. So once this fails, then we get cancer. So how this cancer cell break rules, and basically how they rule the body. So two examples here I have put. One is unguarded cell cycle. To basically example, because I think we are all familiar with the cell cycle checkpoints, how the mitosis happened. So very regulated process is that cell cycle or cell mitosis is a very regulated process, and you can see different checkpoints which regulate each and every phase of the cell division. So in a cancer cell, these proteins, these cyclins or cyclin dependent kinase, and so many thousands of other proteins are there, or factors are there, which regulates a proper guarded cell cycle. In cancer, they get altered, or certain things will get mutated. Then these checkpoints cannot work. These checkpoints, what they do, basically they check for the proper DNA structure, or they look for that DNA is intact, or is there a mutation, or whether there has been proofread. All these things is checked. And if there is any alteration in this checkpoint, that means the malfunction of the checkpoint, the cells cannot go proper proofreading of the DNA before division, and it can give rise to even more mutation, which can lead to altered cellular function, and finally carcinogenesis. Similarly, in our body, the cells divide for a finite time and then die off. That is the rule they have. The apoptosis is the mechanism which a programmed cell death we call, and a cells die off after getting signals, death signals. And which are death signals are again nothing but the proteins which comes from different signaling pathways, and all these proteins which regulate cell death are again evoked in cancer cell. They do not, either they are mutated or they are regulated in a they are down regulated, so the cancer cell will not die off. They will not see the death signal and they will keep on proliferating. This is how, these are the two examples how cancer cell break the cellular homeostasis and they get into carcinogenesis. So this is a depiction from how the gene alteration leads to tumor formation and tumor to malignant tumor and then become cancer. So this is a pictorial depiction for colorectal cancer, which is well studied one. If you see, these are the epithelial lining which has normal epithelium. The first thing which get mutated is APC. If APC is a gene which is very critical for Wnt signaling. Wnt signaling is at early developmental pathway signaling. It maintains the intestinal linings. It maintains the cell proliferation, the stem cell population, everything in the intestine. And once this APC get mutated, the cells become adenoma and they become dysplastic, right? And then you can see there is a proliferation of the cells upon this mutation. Then the next hit is KRAS. KRAS is a very critical regulator for cell proliferation and cell growth, and it is very important for signaling like EGFR signaling and all. And again, further growth happens and it becomes benign. It's become a benign tumor from crypt adenoma to benign tumor. And further TGF beta signaling alteration like SMAT4 mutation and T53, which is, we call it the guardian kind of, it's a master regulator of the cell cycle or cell death. The P53 gets mutated and the cell finally turns from late adenoma or hyperplasia to malignant cancer or malignant tumor. That the cells have now the ability to cross the bridge and they can cross their boundary and they can further metastasize. Now they can spread. So this is how genes regulate each and every phase of tumorigenesis. This is one of the example. Almost all cancer which we see right now are regulated by genes in this, not exactly in this manner, but yeah, somewhat in this manner. There are different genes which get mutated up in a period of time and finally give rise to cancer. And it is not again only mutation. There are other hypermethylation, as I was talking about, epigenetic changes, and there are loss of chromosome is here. So there are a lot of abnormalities which happens, like loss of chromosome, acetylation, methylation of the DNA, and so on. So which combinatorily they give a altered gene regulation. There is a malfunction of multiple genes which leads to carcinogenesis and cancer. So this is how recently all the research have shown what are the characteristics of the cancer cell. So a lot of hallmarks has come out, as we were talking. They can proliferate uncontrollably. They do not die open growth, they evade the growth suppressor. And one of the interesting things, they also avoid immune destruction. Our immune cells are pretty much capable of tackling the cancer cell, but the cancer cell, they mutate in such a way, they alter their signaling in such a way that they fool this immune cell very nicely, and thus the immune cell cannot, though they come for surveillance, they cannot attack these cells, cancer cells, and they will just leave them. So that is how the cancer cell outsmart them, and then they grow, they do not die off. Then they get how the cancer cell keep on growing. They get the telomerase enzyme, which is one of the thing in it is there, which keep on in normal cell, it will shorten after certain cell cycle or certain division, but in a cancer cell that keep on getting replenished. So if you have people are trying to target this telomerase inhibitor in order to suppress the tumor proliferation and thus increase the cell death. Similarly, anti-inflammatory drugs are in market now, and it is in clinical trial to target tumor promoting inflammation, which it can target. And angiogenesis is another hallmarks of cancer, where a cancer cell induce new vascular formation. It induce formation of new blood vessels so that it can take more and more nutrient for it to grow. And it can take maximum energy for itself, where the whole body starve to death. And that's why we see a drastic reduction in weight upon cancer in people. Like suddenly they reduce weight because the whole energy is going to the tumor, whereas the rest of the body is not getting enough of the energy, because the tumor is keep on giving getting more and more blood vessels which are fueling it all glucose and all the energy, whereas other cells they stop to death. And as I had mentioned, resisting cell death, genome instability, and their metabolism is also very much altered. So right now researchers are targeting all these hallmarks of cancer, the characteristic of cancer, what genes they regulate, these all hallmarks, which all genes which regulate these each hallmarks. If we target them, can we inhibit the cancer formation, or can we inhibit its growth? So these all are in clinical trials or in clinical practice, the selected targeted inhibition, which reduce the tumor burden and it suppress metastasis and so on. So coming to the complexity of this, when we hear the cancer is, cancer, although a lot of studies has been done, we have improved a lot, but still cancer is one of the most deadly disease among all systemic disease. Why? Because cancer is not a single disease. It doesn't affect one signaling pathway. As you can see from this complex diagram, this is the pathways in cancer. So these are the signaling events which control cancer, progression, onset, everything. So if there are one gene, if get altered, you can see there are so much of crisscross talk, and one gene alteration can lead to a chaos, a havoc it can lead, and thus altering so many other genes. It's just like dominoes theory, where one falls and then it starts a cascade. Like it keep on, one gene gets altered and then it affects other gene, and then so on, so on. And so whole system get into problem, and multiple physiological as well as cellular bioprocesses become defective, which leads to a total chaos and carcinogenesis. So what are the recent advancement which has happened? So here I have taken out 3 recent advancement along with 3 major pillar of chemo. When it comes to treatment, still surgery, chemotherapy and radiotherapy are the 3 major pillars in almost all cancer treatment. Apart from that, there has been a lot of advancement in modern chemotherapeutics, as well as new therapy regimens which are coming up, and these are in last 10 years, in last decade. These are the 3 very important things which has come up. One is Gardasil 9, which is HPV vaccine for cervical cancer. We have a vaccine right now where the women can have this vaccine. This is against human papilloma virus, which causes cervical cancer. So this is a huge leap which has happened, that now we can take vaccine and can be protected from the cervical cancer in females. In 2015, the FDA has approved oncolytic virus therapy. So this is very recent, and how this researchers, the scientists have engineered a certain virus so that this virus can go inside our body and target selectively this cancer cell, and they can just lyse the cancer cell, they can kill the cancer cell. So basically these are engineered oncolytic virus, so they will not affect the normal cells, but they are very, very targeted, they are very, very specific to cancer cell, and they will kill only cancer cell, which is not true in case of radiation and chemotherapy. In radiation and chemotherapy, we see, and many of you would have seen, a lot of patients, they do have a lot of side effects, which is one of the major drawback of cancer therapy. So maybe in future, this type of treatment like oncolytic virus therapy, which can target specifically to a tumor by not affecting other neighboring cells which are healthy. So we can drastically reduce the side effects which we get from chemotherapy or radiotherapy. The third one is very interesting, which got approved in 2017. These are chimeric antigen receptor T cells therapy, CAR T cell therapy. And this what happens in this case, the T cells are isolated from the patients which has the cancer, and then they will be grown in the laboratory by engineered. They will be engineered in such a way that they can target this tumor which is present inside the same patient, and they will be cultured in this laboratory in a huge amount, and they will be injected back into the same patient. So this is like our own system is being boosted to a better, stronger agent, so that they can fight for themselves. So here we are fighting ourselves. Basically, there has been no outside chemo or radio regimen or any kind of virus or any bacteria. It is pure our own T cells are fighting for the tumor, obviously with the trained genetic engineering. So these are the three advancements which has happened in last 10 years. And so let's next come to the environment and the cancer. So environment plays a very big role in cancer incidence and progression, because as we have discussed previously, the environment has a lot of factors which we know few, and a lot has to be figured it out. We do not know a lot what are the factors which are carcinogens, what are the factors which damage the DNA. A lot has to be found. So what are the risk factor? Majorly I have categorized into three things. Carcinogens are the factors, let it be chemical or any things which are from the environment which you get, let it be pesticides or pollutants or anything which can cause cancer. Any chemical which can cause cancer or carcinogenesis process, we call it carcinogen. Then second one, very critical, which has been brought out previously in this meeting, is the diet. How important is the diet? And it is one of the theme of this webinar, is the diet or nutrition. How critical is diet in cancer management, we will be talking about, and obviously the pathogens which comes from the environment, which also directly can cause cancer. So one by one we'll go. So how the carcinogens works? Basically, the carcinogens or the chemicals, they can damage the DNA, and a faulty repair system, with a faulty repair system, they cannot this DNA will be repaired in a very faulty way without proper proofread. So basically a lot of mutation can be accumulated, or it can also give rise to altered protein function, which finally can give rise to carcinogenesis. Other way, the carcinogens also can be cytotoxic. In that way, they will not damage the DNA, but they will damage the cellular cytosolic proteins, and in turn the cytosolic proteins get damaged, and they can also damage the DNA or damage repair processes or DNA stabilizing protein which are there. They will be damaging, and further this both can lead to carcinogenesis. These are the two ways the carcinogen works. So these are the list. This list is very long, but here I have just selectively put out few which are known carcinogens which we get from the environment. And one of the interesting thing I found is aflatoxin, which comes from a mold. I think it's you guys know better the Aspergillus parasiticus or Aspergillus flavus, which is a mold, and this infect the peanuts, and these peanuts, they can have this Aspergillus, they have this aflatoxin B1 toxin, which is a carcinogen. It can cause liver cancer directly. And also this plants, if animals are feeding on that plant, they can also have aflatoxin M1. It's a different variant, and that also in turn can cause liver cancer, inflammation, immunosuppression and all. So this is a mold which is a carcinogen, which can be found naturally. And similarly, benzene and butadiene, which we found in smoke, which is one of the leading cause of lung cancer related death, smoking. And it is it is very well known how this has been a deadly agent for the carcinogenesis, and how we should stay away from this particular thing as much as possible. And the secondhand smoke also can cause cancer. And obviously the UV radiation, which is one of the well-studied one, how they damage DNA and finally give rise to mutation. And obviously nonetheless, these pollutants which we are keep on producing because of the industrialization and improper management. This all pollutants, there are a lot of factors, thousands of factors which are uncharacterized, which can be carcinogen and which can cause cancers. So here are the 4 things which environmental research we should be focusing on. What we should look for in present and future in order to make a better environment, and which in turn can keep us healthy, or it can have a sustainable way to live in this planet. So first thing, identifying radioprotective components from natural. This is I think the research potential is very high in this, because if we can find out radioprotective components from natural sources, say for plant products or some any of the herbs or anywhere, we can find out certain radioprotective element, that would be really great for medicines, as well as so many ectopic ointment and all, which can keep us protected from all the radiations which we exposed from the sun, as well as various other industrial agents. Second thing, we must look or find out ways to improve the soil quality for agriculture, because this is what we do, because the overwhelming population is forcing us to go for all these pesticide based and fertilizer based thing, and we as botany student, you guys all can aim for finding out new ways, and how we can improve the soil quality by maybe some organic thing, or by any means, out of the box, which is very cheap or possible, a viable way of improving the soil quality, which can be a great fit for better agriculture and better health. Third thing is improved waste management by bioremediation. This has a huge future too, because the waste is keep on increasing with the population. The amount of waste we are producing in the whole world, and in country like us in India, we do not have proper waste management system. And you can see anywhere around you that there are a lot of waste which are there, which has to be managed properly, because those waste can lead to different diseases, not only cancer, because exposing to those exposure to those waste component can be really detrimental. So we should find out, or we should look into research. We should be looking for new methods where we can manage the waste, such as like processes like bioremediation and all, and efficient and affordable bioenergy, which is there for quite a long time, but we have not been able to find efficient ways of doing it. But the research is still going on. A lot of improvement is there, and there are a lot of examples around us where people use or bioenergy and all. But we have to do been better than that in order to keep a sustainable environment. And overall, I believe by keeping all this, if these all pillars go together, all these branches go together hand to hand, then we can have a better environment, and obviously that would impact on our health in a better way. So the next important component is diet and nutrition in cancer, which is a huge topic of research recently. People are talking about how diet can influence cancer onset, as well as it can prevent cancer. So we'll be talking how diet does that and what is the important thing. So this is from in recent NCI data. You can see here that excessive body weight, poor nutrition, physical inactivity, and excess alcohol consumption, it is the cause for 1 in 5 cancer cases. So you can see how much this impact, the food what we take, how much it has impact on cancer onset or carcinogenesis. And overweight or obesity raises a person risk of getting 1 or more cancer. There are 13 different type of cancer one person can get. If we are not exercising, if we are eating lot of calories, we are obese, then our risk of getting cancer goes by multiple fold. So recent research have suggested that we should eat more kind of fruit and vegetables, not and try to reduce junk food, such as highly processed food, red meat, which is a big risk factor, at least in colorectal cancer, although there has been no direct link in other cancer, in intestinal cancer, red meat has been a big risk factor. So consumption of a lot of red meat and highly processed food can really change your metabolism, and thus it can impart lot of metabolites which can impact in a very negative way to your cellular processes and give rise to cancer. And alcohol, no need to talk about it because it has got lot to do with liver inflammation and liver cirrhosis, and finally liver cancer and other cancer also. So it's best to avoid alcohol as much as possible. So how the diet works inside a cellular system, how this diet, we are taking so many different type of food which has got different multiple component, carbohydrate, proteins, fatty acids, we are taking a lot, and how this each and every component of those nutrients plays role in molecular regulation. You can see antioxidant which are there in a lot of vegetables and fruits, we can see, and this antioxidant, they really consume all the free radicals which are produced in our body, and they consume it and thus protect the protein function and protein structure, and thus keep a healthy cellular homeostasis in our body. There are a lot of secondary metabolites from flavonoids and which has been shown to regulate multiple cellular regulatory pathways such as MAPK pathways, PI3K pathways. These are all signaling pathways which are very critical for cancer proliferation and growth. So these flavonoids which you get from these plants or plants product are very critical for regulating. They have direct regulation over this molecular signaling, which is very critical. And polyunsaturated fatty acid, this fatty acid has a lot to do with inflammation. Lot to do with inflammation. It's a coming of hot topic of research nowadays, how these fatty acids are causing other obesity, fatty acid, and how the cancer inflammation is linked. So this polyunsaturated fatty acid, they do influence different level inflammation process at different level, including NF-kappa B downregulation and all. So NF-kappa B is a pathway which regulates the inflammatory process inside our cells. Red meat, processed meat, as I spoke just before some time, it is like induced pro-carcinogen, and if we must reduce this. If you reduce the red meat and processed meat, you keep your intestinal integrity. You reduce the pro-carcinogen if you reduce the intake of red and processed meat. And this red and processed meat has a lot to do with probiotic bacteria which are sitting in our gut, which I'll be talking in the next slides, how the gut bacteria which are there inside our system can modify the digested component of this red meat and processed meat, as well as dietary fibers. If you take fiber rich food, then how a gut bacteria respond, and they change into a better way, or they produce better secondary metabolites, which in turn plays a good role in maintaining cellular homeostasis. So this is one of the booming area in multiple systemic disease right now, including cancer, and it is least explored because this interplay is very complicated. So what we eat is directly influence the host metabolism, and this diet is directly linked to this gut microbiome. Gut microbiomes are the bacteria which are sitting in our intestine. Okay, there are millions of trillions of bacteria which are sitting in our intestine, and we don't know them yet. It's a big way the research is coming up, how this gut biome changes almost all aspect of our cellular physiology. And this is already known to, there are certain gut bacteria which are already known to prevent as well as cause carcinogenesis process, although not directly but indirectly. Yeah. So let's coming to biome. So when we are less human than we think. Why? The answer is over here. The bacterial cells in our colon is about 40 trillion, and human cells, we have 30 trillion total body cells. So we have more of bacterial cells than human cells inside our body, and we do not know yet what these bacteria they do, what all these bacteria are, what are their function, why they are there inside our intestine. So it's a huge area of research, and they regulate multiple metabolism, and they produce a lot of secondary metabolites which are still majorly uncharacterized and their function. So, but recent research have shown that how this prudent diet, the diet rich with vegetable and fruits, is that will produce short chain fatty acid, which in turn are basically byproduct of this bacteria. So once you take this prudent diet, this dietary factors goes into your intestine, and then this biome which is present in your intestine do some chemical reaction, they process that your dietary factors and they produce short chain fatty acid, which is very, very good for your boost of your immunity, as well as anti-carcinogenous product, and which maintains a very healthy cellular processes and cell homeostasis. Whereas the Western diet, which is rich in fatty acids or high fat diet, which has been shown that this bacteria get affected, they produce different type of metabolite than here, and which are pro-carcinogen. They will enhance the process of carcinogenesis. They'll reduce your metabolism. They'll reduce your immunity. They increase the inflammation, thus causing cancer and various other systemic diseases. So this is something very, very interesting and coming in a big, big way, that what this gut biome does inside our cells, inside our body. The third component which comes from biology in environment is biological agents. So obviously, although there are viruses, bacteria which can directly cause cancer, which are known like Epstein-Barr viruses in lymphoma, hepatitis B and C, they can cause hepatocellular carcinoma, human papilloma virus, they can cause cervical cancer, and as I had discussed previously, there is a vaccine available right now for human papilloma virus, and we all where the female can be protected from getting this particular cancer if vaccinated at a proper age. And Rajiv Gandhi Center for Biotechnology actually runs in Trivandrum one of the biggest this vaccination study on human papilloma viruses in India. And bacteria, there are a lot of bacteria which has been found, and here I have just listed out few. Helicobacter pylori is very known deadly pathogen which causes gastritis as well as gastric cancer. And few studies have shown Salmonella and Salmonella typhi to be associated with gallbladder carcinoma and colon carcinoma. Although these two are not directly, there are no evidence where it can directly cause cancer, but obviously it can, it triggers, or it can accelerate the process of carcinogenesis. Whereas the Helicobacter pylori is enough to cause gastric cancer alone, without any mutation or without any further gene alteration. So basically, how this virus and bacteria, they do cause this altered gene expression or inside our host cells by gene transfer and producing a lot of genotoxins which can damage the DNA, and obviously a lot of secondary metabolites which can be carcinogenic in nature, and they can cause cancer. So I thought I would keep a few slides on plants because it's a department of botany is hosting this event. So I thought it would be good to see what are the aspects which we have for students of botany or those who wants to pursue their career in future botanical research and in cancer background. Obviously, there are so many other things which I don't know, but I'll be talking about what the importance of plants in cancer treatment, at least, because there are a lot of plant metabolite or plant secondary metabolite which are in clinical trial. A lot, you can go through literature and it's a never-ending list which is in various phases of clinical trial. The secondary metabolites are there, but here I have put out three components which are very, very famous and already in clinical in clinical use, like vincristine for Catharanthus roseus, and which is treated which is a drug frequently used in leukemia treatment, and from Texas brevifolia paclitaxel, which is a very famous anti-tumor agent discovered from the plant, this particular plant, which is treated not only in breast cancer, head and neck cancer. The list is even longer, but I have just highlighted these two different cancer. So these two plant products, basically they inhibit the the microtubule function in cell cycle. So not in cell cycle, in cells. So basically in cell cycle, when cell divides, the microtubule is very, very important for the chromosome to separate. So these two Christian pancreatic cell, they act on microtubule and they inhibit the cell to grow. So basically they target highly proliferating cells, which is one of the hallmark of cancer. And thus these two are very famous chemotherapeutic agents which are in use, and they are purely discovered from the plant. And one of the things which we know, we all would have heard, is the curcuma longa, the curcumin, which is a famous thing which is we use in our country. In India, it is used pretty much everywhere. The curcumin, the turmeric, and that one of the most important component of the turmeric is curcumin. It is in clinical trial right now in multiple different type of cancer, that it has got a broad range of action in different signaling pathways. It has been shown to be very, very effective in multiple cancer treatment in early clinical trials. Hopefully this would come out as another magic drug in future cancer therapeutics. So what are the challenges we have as botany research, or being a student of botany? The challenges which we have is identification of pure active compound from plant. If we want to identify, it is always the challenge is big, that to identify the active component inside the let's take any part of the plant, and if you think it's a extract, then you must identify a pure compound in order to make it more effective, because and it's that process of extraction is very, very tedious and time consuming, and the processes are also very, very expensive, and always people have shown that the yield is very, very limited. So that all these challenges also opens up opportunity for us, which I believe that the diversity of flora in our country and the knowledge which we have, we can use in identifying better novel compounds from this plant. And I think we can purify better to get better compounds. And one of the things which I find very interesting is that people have shown, and you can go back to literature to see, what they are doing is that they are genetically engineering plant to improve the metabolite production, because the less yield is a big challenge. If you do cultivation of particular some anti-cancer producing agent producing plant, and the yield is very less, what people have done is that they are genetically engineering the plant, so that the plant produces much more secondary metabolites than usual. So this is something very, very interesting. And using this knowledge what we have on our flora, and how the production can be improved, how the agriculture of this plant can be improved, can really change the way we look at discovery of new cancer therapeutics in future. So I have put here 4 pillars to fight cancer better, which I believe. The first one is obviously the basic biological research. Let it be any field, let it be botany, zoology, all this field, chemistry, physics, all should come hand in hand in order to fight, in order to come out with new novel ideas of purification, or devising an instrument which can purify the things better, or to understand the cell function, how the cancer cell works. All basic biology is very, very critical in order to, these are like the base of any innovation and any discovery. So that is one of the major things. And second thing is translational research, because we cannot keep on looking at basic research, because the cancer cases are increasing. So we cannot let people die just because we are understanding the basic biology. So I believe the translational research should go hand in hand in order to fight the cancer better. So we should have basic research, but we should also have translational research, where we are looking to find out very rapidly are trying out new compounds in clinical trials or early clinical trials, where we can just put them into patients so that we can have new novel compounds at the same time. The third thing, very important, sustainable environment. The environment is keep on degrading. Nothing, we all have experienced it in the past 1 year, or what COVID has brought. How the environment, we could see the change in environment because of lockdown and all. So how much damage we are doing to the environment is pretty much clear for us. So we must work together to have a very prospering environment, very healthy environment. So the research should also focus on how to have a very sustainable environment. We should be productive, we should be able to produce, agriculture should grow, industry should also has to work, but at the same time we have to protect it from the environment also. So we have to come up with new ideas, novel methods in that way. And the fourth thing is very critical in cancer fight is public awareness. I believe that if we all get educated what to eat, how to exercise, and we know how early diagnosis protocol, and when we should go to clinics, we should frequently visit the doctor if we have something. Those kind of awareness is very important in order to fight cancer, because if a cancer is detected at a late phase, it is very tough to fight. So only public awareness on cancer can enable us to get a patient diagnosed earlier. Okay, so I believe the public awareness is very important, along with all the other field of research. So these are the few interesting facts I have put out for students to go and they can read on it if they find it very interesting. That we do not, nearly our heart rarely get cancer. It's nearly immune to cancer. Why? It is very interesting, because most of our organ get cancer. Why heart is special, and why elephants are so big but they do not get cancer, or they get cancer but it's very rare. They are huge animal, but how they are protected from cancer. These are the same thing. And few recent discovery like cell cannibalism in cancer cell, how a cancer cell eat another cancer cell, and what they do, whether it is a protective, whether it's a cell killing mechanism, or whether they protect each other, is a very beautiful coming up area students could look for, and they can study if they want. I have put it here. And thank you for your attention, and I stop here. And as I always put a quote from Siddharth Mukherjee, the freaks become norms and norms become extinct, and monster by monster evolution advance. So basically the cancer is also kind of, we can say, it's an evolved evolution inside human, which can take us down if we are not careful enough, we are not guided enough. Then we don't know, like maybe we have to lose ourselves to this particular disease, because cancer is something much more advanced. This is something which has evolved inside us, not from any outside matter. So thank you. I'm ready to take questions. Thank you for your attention. Hello.
Okay.
Yeah.
I can. Yes. Thank you, Dr. Tapas. Somebody have asked a question. Surya Ramesh, why genetic diseases cannot be cured?
Yeah. So a nice question, Surya. Genetic disease cannot be cured because you carry a defective element inside you, right? If a genetic disease means that is something we have wrong in our chromosomes, or maybe in our DNA, we carry a faulty gene, so which is very tough to get rid of. So that we cannot get rid of that thing. But mostly what recent advancement, many of the genetic diseases, there are drugs which can control a particular faulty phenotype of the gene. Say for we carry one altered protein in us, we inherit from our parents. So what happens that we cannot do anything about it. A child is born with that faulty protein, but once the child is getting diagnosed that it is having a faulty protein, that what that faulty protein does, it can be targeted by different drugs. And it is a huge market how these genetic diseases drugs are very costly, because they target these particular genes, so they are very costly. But again, you are that is a sad reality, that most of the genetic disease are tough to treat, or in cancer also 5 to 10% of the cancers are genetic inherited. So those cases, what you have to do is you have to get diagnosed early and take drugs, have a healthy lifestyle, and that's all, because we are unlucky in that way. Yeah. Yeah. So another question by Nandana Basis: what is mean by monoclonal antibodies? How can it cure cancer? So, yeah, this is very interesting one. Monoclonal antibody therapy has been, already FDA has been approved. And what are monoclonal antibodies? Monoclonal antibodies are the which targets antibodies. I think you have an idea. We have antibodies, we produce antibodies, our B cells produce antibodies against multiple pathogens or multiple antigens, which gets so basically we have a defense mechanism, the immunity, the immune cells which produce antibody. These are the proteins which targets this antigen and they neutralize it. That is antibody. But then what is monoclonal antibody? Monoclonal antibody is that the antibody which binds to a particular epitope. Epitope means a antigen can multiple epitope. Epitope in the sense antibody binding sites. So antigen, say for antigen, some viral protein is coming to your, say for COVID protein is coming, COVID virus is coming. So virus can have multiple, that COVID virus can have multiple epitope on it. But if our immune cell produce a particular antibody which is for a single epitope, that we call it monoclonal antibody. But if a antibody which can bind to multiple epitope on that COVID virus, SARS virus, then we call it polyclonal antibody. And yes, by monoclonal antibody is a huge success in cancer treatment, at least breast cancer, where Herceptin has changed the way we treat breast cancer, which are very resistant to many chemotherapeutic drugs, because this monoclonal antibody can specifically target a single protein, and those proteins are expressed in particular cancer cells. So they can, if you give a monoclonal antibody, they can go and bind to the protein directly, the cancer cell directly, whereas they don't bind to any other normal cell which are not expressing that antibody antigen, sorry. So in that way, monoclonal antibody is a huge success. It is a targeted therapy, monoclonal antibody therapy. Very targeted. It can target cancer cells specifically. Hope I made myself clear. Is there any specific viral particle that can activate pro-cancerous cell? So it is not a viral particle basically, which according to my knowledge, I don't know much, but to answer your question, Arshad, it is not viral particle which induces or it can induce a healthy cells to become pro-cancerous or a pro-cancerous cell to become cancerous. It is the genetic element which they transfer basically. They transfer, this virus can get inside cell, or they can just transfer a genetic element into our host cells. And then like E16, E18 protein, if I'm not wrong, which are very important in I think it's a part of HPV, human papilloma virus. They give this genetic element, and which we do not have. We carry that genes from virus, and that those genes are very, very important in regulating our cellular processes. So although it is from a virus, those genes comes to our cells and they regulate the cellular processes. So in that way, they basically hijack it.
Okay. Thank you, Dr. Tapas. So one more
Thank,
question is
yep.
there. One more question. Why T cell receptor therapy not exploited in our country? Is it very expensive or
Uh,
it is
I
usually
do
used
not,
in developed
yeah,
countries?
yeah, that's true. That's true. That's true. But CAR-T therapy would be very, very expensive as well as. But I don't know personally answer to this question, because I don't know whether in India we do not have CAR-T therapy. Maybe some major hospitals would be practicing it, because this is something I think can be done, because the advancement already FDA has approved. So as you said, it would be very costly. I agree with that fact. Maybe it is costly, but there is no reason why we cannot use it.
Okay. Thank you. One more question.
Yeah,
It's like a twisted question, maybe I can say that.
yeah,
Different types of cancer is much vibrant in youngsters nowadays. So
yeah, yeah.
how is your approach on this?
Yeah, so that is a very nice question, and actually, I was part of that kind of study, that young population where they are getting cancer in Kerala. Basically, we used to study that. So the approach which I spoke in this particular presentation is one of the factors is that our lifestyle, which has changed a lot, and maybe we are exposed to more of the carcinogens, unknown carcinogens in our daily to daily life. So basically, although we do not carry a inherited faulty gene with us, what we are exposed to very bad, maybe carcinogenic food in earlier our life. Very early, we are eating a lot of junk food, and we are basically, as the technology and advance has advanced, we are not exercising much more the way we used to do. We are not walking anymore. We are just going by bike, or we are going by car. So physical inactivity is also one of the reason. And I think to fight against this young cancer domain, we must aware young students, young people, to be vigilant about their exercise, their food. They should take more of, like I had mentioned in my presentation, fruit and fiber rich food rather than junk food or any other, high fatty food. So I think that would be my message. Although the biology is not properly studied, it would be really interesting to look in research, further research, what causes this young question. But I would say these are the points which we should take home to fight.
Okay, thank you. One more question. It's like a twisted, again, it's like a twisted question, or it's a irony, I can say that. So even some of the vegetarians who choose healthy lifestyle, we can see cancer is there for some youngsters. No, in vegetarian people, even though they choose a healthy lifestyle.
Yes.
Why is it so?
You mean that even though they follow a healthy lifestyle, they
Yeah,
eat
in
vegetables,
vegetarians.
they get cancer, right?
Yeah.
Yeah. So if you see earlier slide of my presentation, you can find there are 2 points I had mentioned. One of the reason is that faulty replication. Okay, there is something called faulty replication. So daily to daily we have replication error in our body. So we don't know if our replication process is not 100%. Some means the replication proofread is not 100%. So there is a possibility that sometimes the replication process during the replication, the cells, the DNA is mutated in a gene which is crucial for the cell cycle regulation or other processes. So I would say, and with aging, another factor. With aging, say for once we age, our whole system get aged, right? It is not like that our body is cells are not aging. Our body tissue systems are also aging. So their efficiency reduces. Once the efficiency reduces, whether you are a vegetarian, whether you are a vegan, whether you are a non-vegetarian, there is a possibility that you might end up accumulating mutant protein or faulty genes inside you, which can lead to, yeah. But that's just, you are unlucky. That's all we can say. You are unlucky to get, even though you're following. Yep.
So again, a question comes uh, like uh, uh, like T cell receptor therapy. Like here in developing countries like India, prognosis studies are not happening, but diagnostic studies are happening. Likewise, prognosis studies are much vibrant in the case of developed countries. So, what is your approach for that?
Diagnosis and prognosis both are actually, we are lacking, we can say. It is not like the diagnosis is very good and prognosis is not here in our country. We do not have awareness, as I told, public awareness. So people have some problem, they do not go to doctor for, they do not go to, their scare of cancer, which we should not be. A message: if you have any problem, just walk to a doctor and talk regarding your problems. Just be open with your parents. Whatever physical issues you have, just open up with your parents. That is one of the major reason why we do not get diagnosed early in our country. It is not like that. At least states like Kerala, you have a very, there is, the health system is pretty good compared to other part. But still people hesitate to go. So abroad, the awareness is much more better. They have better, obviously reachable diagnosis thing, and prognostic thing is there, because they can spend a lot of money in different clinical trials or clinical studies, which we also do. But I think the major part comes is the awareness. If we get aware, I think we have everything in our country also to diagnose and prognose better.
Thank you very much, Dr. Tapas, for this informative session. Good news
Thank
is
you.
that every plant science students or students from any other branch of science can opt for cancer research.
Yeah,
Actually,
sure. That was my purpose of, it's of this presentation. At the end, what I suggested about environment, you guys, botany student, huge role. Do not, it is not about learning genetic engineering and always, or some cell biology. The botany, as I told, we have lots of flora to investigate, lot of compounds to look for, and which can really turn out, you can end up in discovering new compounds for cancer treatment. And always the environment, which you guys can only lead us, right? Environment is a huge factor, and the student of a plant science student only can lead us in having a very sustainable environment and a very healthy environment. So I suggest, yeah, all fields should come together, and to be motivated enough to find a solution for all the problems we have. Yep.
so actually, students have a lot of opportunities to select their career. It's just that timely action and proper planning is needed.
Proper, yeah, guidance. Yeah, that's true. That's true.
Once again, thank you very much. We just come to an end of session 1. Let's start session 2.
Thank
So
you.
I would like to we can move into session 2. We would like to introduce our next keynote speaker, Dr. Aneesh Kumar Asokan. Dr. Aneesh got bachelor's degree from University of Kerala and master's degree from Jawaharlal Nehru University, New Delhi. He did his PhD from Rajiv Gandhi Center for Biotechnology, Trivandrum, on the topic identification of the altered metabolism in individuals at risk for type 2 diabetes mellitus through mass spectrometry-based metabolomics. He was awarded for Newton Baba PhD placement at University of Birmingham, UK. Actually, there are lots to talk about Dr. Aneesh from his bucket list, but I would like to describe him in one sentence. A person with his limited facilities, he just went limitless. Dr. Aneesh will be talking about vaccination and its importance in public health. A massive vaccination program is going on against the COVID-19 throughout the world in an unprecedented manner. However, primary education in vaccine lacks for the public. The information related to vaccines, such as definition, mode of action, types, clinical trials, effectiveness, herd immunity, history of immunization, types of COVID-19 vaccines, and common myths will be presented. Now, I invite Dr. Aneesh for the presentation. Over to you, Dr. Aneesh.
Yeah, thank you, Surekha, for a nice introduction. Actually, I will now start sharing my
Share.
slide. Okay. Okay. Wait a second. Yeah, can you see my slides now?
Yes, Dr. Aneesh.
Okay. Yeah.
Yes.
Okay, we'll start now. Respected principal and other teachers and my dear students, I will be talking about the subject vaccination, the cornerstone of public health. Actually, why I'm talking this topic now, you all know we are going through a very difficult situation in the entire globe, but at the same time, we are seeing a massive vaccine program around the world. Means ever the world is never see that many number of vaccination, but the same time some minority of the people, they are against the vaccination. uh, for example, a news came in the last week in India Today news. Around 60 percentage of the polled Indians still hesitant towards COVID vaccine, showed by this survey. This survey was conducted in 17,000 people in different part of India. So this is a very bad news for us, especially for healthcare system. It's not only in India, in abroad also. In USA, a study conducted in 2010 shows that around 3% of the parents, they refused all vaccines for their child, different reasons. And this is not about without knowledge. Actually, this number is much higher in certain communities, especially states that have laws allowing more exemptions. And now a new term came in our dictionary called vaccine hesitancy. It's a term to describe the range of feelings parents have about vaccine concerns. So WHO defines it as a behavior influenced by number of factors, including issues of confidence, people not trusting vaccine or their provider, and they are not perceiving the need for a vaccine and do not value the vaccine or the accessibility or access to the vaccine. This we called vaccine hesitancy. And we can look why are parents worried about vaccination. And there are several reasons. First one is maybe religious reasons. Like some people thinking a vaccine contains some of the animal products. They are against their religious belief. And there are some philosophical and personal reasons. These are mainly people are believing in the natural remedies. They are thinking the natural immunity is better than the vaccine. And another one, safety concerns. Parents are thinking the vaccine can cause autism or it can cause infections or side effect. This is another way, and we will talk about later. And some people are not means they need more information, and in some people even they are not trusting vaccine at all. And why this is happening, especially in these days, the recent times, because the one reason, disinformation on internet, especially in the social media, WhatsApp group and Facebook. So people are not evaluating the risk of disease versus risk of vaccine reaction. So vaccine reaction may be a mild reaction compared to the disease itself. And our physicians or nurses, they have the duty to convince the parents. They also lacking the a proper knowledge to convince the parents about the vaccination. And another thing, this is the because of vaccination, some of the deadly diseases such as uh, polio, smallpox, which are disappeared from our community. We are not seeing that disease now. So we are forget the importance of vaccination because of lack of those disease. And another one, lack of altruism. That means altruism means concern for others. Actually, when I am getting vaccine, it is not only about me. It is about us. I'm not only protecting me. It is I am protecting my community. That feeling also has to be come in people. But that also one reason. And some of the myth I will talk. The major, maybe you heard about this vaccine cause autism. Actually, this thing came in early 1990s. One study published from England, which shows that some vaccine causes autism in children, and this made some big news in those time. And means the later checking shows that this was like a manipulated study, and the paper was retracted, and the lead author had his all license revoked, and subsequent studies have shows no connection between vaccines and ingredients with the autism. So I can clearly say that till now there is no link exists between vaccines and autism. And I will talk about some of the myth, especially regarding the COVID vaccine. So the first question will be COVID vaccine can affect women's fertility. No, the fact is that it not affect fertility. Actually, getting COVID, other hand, can have potential serious impact on pregnancy and mother's health. And another myth: if I have already COVID-19, I don't need a vaccine. No, there is no enough information currently available to say if for how long people are protected from COVID-19 after they have the disease. So we don't know how long you are protected from COVID-19 after the infection. The other misconception: getting COVID-19 vaccine means I can stop wearing my mask and taking coronavirus precaution. No, you cannot stop the all precautions, because vaccine cannot stop the entry of coronavirus in your body. This only prevent from developing moderate to severe COVID infection, and still you can act as a carrier and you can give disease to your close people. So you should follow the current prevention protocol even after the vaccine. And another one, side effect of COVID vaccines are dangerous. No, actually, we cannot call it side effect. Actually, it's the effect of vaccine. Maybe after vaccination, you will have some pain in the injection site, maybe body ache, headache, or fever. That may last for a day or two. And this is a sign that vaccine is working in your immune system. And if the symptom is persisting beyond 2 days, you should call your doctor. Otherwise, it is a natural process. And another one, the COVID-19 vaccine enters your cells and change your DNA. This is especially regarding the mRNA vaccine. Actually, mRNAs are highly unstable molecule. They will quickly break down inside the body without affecting your DNA. Another one, COVID vaccine was developed without or contains controversial subject. Actually, the vaccine contains, if it is an mRNA, it's contain mRNA or subunit vaccines that protein and some salt and some sugar. Means we have to make the injecting solution very similar to the blood regarding pH and ionicity and all. For some, so we have to add some salt and sugar. Otherwise, it is not containing any fetal tissue or materials such as some implanting implants, microchips, or tracking devices. These are media constructed news. And this immunization or vaccination is not a new treatment in our population. Actually, every year around 3 million kids are saved due to immunization. Vaccines are able to protect against the following 26 infections, with many more more in development. Example: cholera, dengue. The list is a long list. So we have a long history of vaccination. And okay, I told so many things about vaccine. So to refresh your memory, I will talk about what are vaccines. Vaccines are attenuated killed microorganisms or protein derived from administered for the prevention, treatment, or amelioration of infectious disease. These are some textbook definition. Actually, the basic concept is like that. Our immunity have 1 minute. Hello. സുരേഖ പറഞ്ഞു. വല്ല ഓക്കെ ഓക്കെ ഓക്കെ. ഇപ്പോഴോ? ഇപ്പൊ ഓക്കെ ആണോ? ഓക്കെ. Yeah, I'll start once more. Like, ഇപ്പൊ കറക്റ്റ് ആണോ? Hello, can you see the slides now in full screen mode? Hello? Hello? Is it correct now?
Yes, 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 but it's not in full screen actually.
Now also?
Yes, yes, it's not in full screen.
Now?
It is not happening.
Now, like which slide of vaccine hesitancy, that slide is in?
Yes, decreasing vaccine rates. Yes, vaccination rates.
Okay.
Okay.
Okay, and now the slide is immunization saves lives and keeps people healthy. Okay, should I continue? Like, can you see now?
Yes, we can see the slides. You can continue.
Okay.
Okay.
Yeah, actually some issue. Like I
Yes,
don't.
yes.
Okay, should I continue now? Like,
Yes, you can make. Yes, you click that on that or F5.
okay.
Function 5.
Now also not in full screen mode.
No, it's not in the full screen mode.
Okay, I will
I
look.
think you have to click on it, right?
Means where I have to click for that full screen?
Like the usual PowerPoint presentation, you have to click. Function 5. Yeah.
Function 5.
Or maybe it is not happening.
Now. Okay, I'll like.
No problem.
Like I will share once more.
Yes, if it is remaining like that itself, then it's okay.
Okay.
You can continue on it.
Okay, now it is in full screen
It's,
or
it's okay. It's okay. You can continue with the third slide.
okay. Yeah, now we can see the vaccination rate slide.
The first slide. The first slide we can see.
Only the first slide. Okay. I think okay. Slides are not changing now.
Yes, now it's changing. As you're clicking on it, we can see the screen actually.
Okay, means I had to add the full screen. Okay, how to do that? Actually, don't know. Hmm. Okay, I think I
Full
will
screen.
continue the presentation.
അവരുടെ ഫുൾ സ്ക്രീനേ ആ സാറിന്റെ ഫുൾ സ്ക്രീൻ ഇങ്ങനെ പവർ പോയിന്റ് ഫംഗ്ഷൻ F5 തന്നെയാണ്.
PowerPoint open ചെയ്തിട്ട് F5 ഓ?
Yes, yes. The usual mode only.
Now? Is it fine now?
Aneesh, go to slideshow. You will see a drop down.
Slideshow, okay.
Slideshow. Then drop down. You will see a drop down.
Drop down.
From
Present
current
on
slide. Current slideshow. Click on to current slideshow.
Okay, from current
From
slide.
current, yes, yes, from current slide. Second one, second one.
Now?
Second one, yes, correct. From current side. Okay. Let's see what.
Now we can see the slide? No.
You maximize it, maximize it. Maximize it. Yeah. Maximize it.
Okay.
Yeah. From current slide.
Now we can see? No.
Yes, now it's happening.
Okay, okay.
From
Okay.
current slide.
Done?
Okay, you can continue.
Okay. Okay, I talked about common myth. Maybe you saw the slide. Now you can see the slides are changing.
Please.
Okay.
Thank you.
Okay, so I will talk about vaccine. So what are vaccines? This is like a typical definition of vaccine. It's like a killed or attenuated organism, but the basic principle is like that. Our immunity is mediated by two different type of cells, especially white blood cells. They are called B cells and T cells. And B cells, which can secrete molecules called antibodies. This antibody can bind to a specific foreign substance. For example, proteins in the virus, we call this antigens. This antigen antibody reaction will be like a lock and key mechanism, means each there is has a highly specific reaction. So in our body we will have a immune cells, B cells, which can bind to the any virus, but the number will be very less. So what vaccine is doing is that when we are giving this vaccine, this contains some protein molecules. This will be recognized by these B cells. The number of these B cells will be increased several times, and this some of the part will produce secrete antibody, and some part will, and some will stay in our bone marrow as a memory cells. This is the B cells. And other class of cells called T cells. This T cells will mediate the cell-mediated immunity, means it will destroy the infected cells, and one part will store in the bone marrow as a uh, memory cells. Because of this reason, the second infection will be during the second infection or with the real virus, body can fight against the virus very properly. Or that means we are basically teaching the immune system with a vaccine for a better immune reaction in the future. This is the whole concept of vaccination process, and we will use different methodology in different vaccines. And this is the mechanism. Actually, we will inject the vaccine. These are the we call this antigen molecules. This in order to enhance the immune reaction, we will give some other molecules called adjuvants. These adjuvants will enhance the immune reaction. The first cells will be the dendritic cells or antigen presenting cells. Will identify this foreign substance, and they will send this information to T cells. T cells are two types, like CD8 and CD4 cells. CD4 T cells, also called T helper cells, which will activate a single B cell. I told the number will be very less, and after this getting this information from the T cell, the number will be increased. We call it proliferation. So one part will become plasma cell. This antibody producing B cells are called plasma cells. They will secrete antibody, and some part will called memory B cell. They will stay in the bone marrow, and some part of long-lived plasma cell also stay in the bone marrow. And the other part, CD8 cells. These cells will differentiate into two types. This is CD8 effector T cell or killer cells, and CD4 memory cells. This cell also stay in the bone marrow for later infection. This is a basic understanding about the physiology of immune reaction during a vaccination. And I want to tell about this COVID-19 more, because this is not like a flu or a fever, because it is not only affecting your upper respiratory system or lungs, it is actually affecting multiple organs in different people. It affects the heart, reproductive system, neurological system. It can affect your psychology, immune and hematology system, urinary system, and digestive system. So it have a multiple organ, high chance of multiple organ failure. So we cannot say that I will be recovered from COVID, and so I will wait for that. So it's not a good idea. We still we don't know how this COVID making complications in a person. And I'll talk about some of the vaccines. These are the Pfizer and Moderna vaccine. These are two mRNA vaccine highly used in US and European countries. And actually, this is the coronavirus. And this coronavirus will have a spike protein. So we are using not the virus, the spike protein for a vaccine, which are the antigens. So we cannot actually, why we are going for mRNA? There is a reason is that a protein-based vaccine needs a high manufacturing process. The manufacturing of protein is not easy as RNA molecule. Means it involves multiple steps, and each step has to be validated by FDA or other regulatory bodies, and this process will take years. We cannot make a protein vaccine within a year, so we went for the mRNA vaccine. So the drawback is that these mRNAs are highly unstable, so we have to store it in a very low temperature. And scientists came with a nice idea. They put the mRNA in a lipid nanoparticle, so it can stay in somewhat stable in the vaccine solution. So when we inject this nanoparticle into a body, maybe we are injecting the muscle. So the muscle will recognize this mRNA by this ribosome, and this will synthesize this red color spike proteins, virus protein, by our cells. And when this cell, and some of the proteins will be expressed in the surface of our cell, and whenever the cells dies, this protein will come out of the cell, and this will be recognized by our T cells, and T cells will give this information to B cells. They are the antibody producing cells. So hello. Okay. Now which slide you are seeing? Yes, slide. Okay. Can you see the slide now?
No, no.
No. Actually, okay. ഇത് ആരെയും ഹെൽപ്പ് ചെയ്യാനായിട്ട് അവിടെ ആരെങ്കിലും ഉണ്ടോ? ചോദിച്ചാൽ എങ്ങനെ ചെയ്യുന്നതെന്ന് പറഞ്ഞു.
അനീസ് സാർ കേൾക്കുന്നുണ്ടോ?
ആ പറഞ്ഞു.
സാറ് PPT നമ്മുടെ ഫുൾ സ്ക്രീനിൽ ഇടാം, ഫുൾ മോഡിൽ ഇടാം.
അതായത് അകത്തുനിന്നാണ്
ഷെയർ ക്ലിക്ക് ചെയ്യുക.
ഓക്കെ, അപ്പൊ ഇത് അകത്തുനിന്നല്ല ഷെയർ ചെയ്യുന്നേ, ആദ്യം സ്ലൈഡ് ഓപ്പൺ ആക്കുകയല്ലേ?
സാറിന്റെ സ്ക്രീൻ ഓൾറെഡി ഇതിൽ വിസിബിൾ ആണ്. സാറ് പ്രസന്റേഷൻ ഫുൾ സ്ക്രീനിൽ ഇട്ടിട്ട് ഈ ഷെയർ ചെയ്തതിനു ശേഷം അത് ഫുൾ സ്ക്രീൻ ആക്കി ഇടുക, എന്നിട്ട് പ്രസന്റേഷൻ മോഡിലേക്ക് ആക്കി ഇടുക.
ഓക്കെ, ഇപ്പൊ ഞാൻ PPT ഓൺ ചെയ്തു, എന്നിട്ട്
ഷെയർ ചെയ്യൂ, ഷെയർ ബട്ടൺ ക്ലിക്ക് ചെയ്യൂ. താഴത്ത് ക്യാം മൈക്കിന്റെ തൊട്ടിപ്പുറത്തെ ഷെയർ എന്ന് പറഞ്ഞ ബട്ടൺ ക്ലിക്ക് ചെയ്യുക.
ഷെയർ എന്നുള്ള ഓക്കെ, ഷെയർ സ്ക്രീൻ.
ഷെയർ സ്ക്രീൻ. അതിൽ എൻ്റർ സ്ക്രീൻ കണ്ടോ ഓപ്ഷൻ?
ഷെയർ സ്ക്രീൻ ടിപ്സ് എന്ന് പറഞ്ഞു വരുന്നു, ഷെയർ സ്ക്രീൻ വരുന്നു. ഷെയർ എൻ്റർ സ്ക്രീൻ, അതെ,
അത് ക്ലിക്ക് ചെയ്യും.
ഓക്കെ, എന്നിട്ട് ഷെയർ
ചെയ്യും.
ഓക്കെ,
ഇപ്പൊ സാറിന്റെ സ്ക്രീൻ ഇപ്പോൾ ഞാൻ ഇതിൽ സ്ട്രീമിലേക്ക് ആഡ് ചെയ്തിട്ടുണ്ട്. ഇനി സാറിന്റെ സ്ക്രീനിൽ ഇത് മാത്രം ഫുൾ സ്ക്രീൻ ആക്കിയിട്ടാൽ മതി, എന്നിട്ട് അത് പ്രസന്റേഷൻ മോഡിലേക്ക് ഇടാം.
ഇപ്പോ ഇതിന് പ്രസന്റേഷൻ മോഡിലാക്കിയാൽ മതി.
ഇത് ഓക്കെ, പ്രസന്റേഷൻ കൊടുത്ത് ഓക്കെ ഫുൾ സ്ക്രീൻ ആയി. ഇനി സാറിന് അവിടെ ചേഞ്ച് ചെയ്യാം. സാർ ഇതിൽ മാത്രം നോക്കി ക്ലാസ് എടുത്താൽ മതി.
ഇപ്പൊ ചേഞ്ച് ചെയ്യുന്നുണ്ടോ?
ചേഞ്ച് ആവണ്ട. ഇപ്പൊ ഡിക്രീസിംഗ് വാക്സിനേഷൻ റേറ്റ്സ് വരെ. ഓക്കെ സർ.
Yeah. Okay. Sorry, actually, okay, there's some technical issues. Okay, I will continue the presentation. So actually, I talked about the this vaccine hesitancy and decreasing vaccination rate. You might saw, maybe you didn't see the slides. Okay, and these are the definition. I explained it. What are vaccine hesitancy? These are some of the slide, and the reasons I explained. And hesitancy is like widespread, and the causes, and some of the common myth. And vaccine is like some of the myth regarding COVID vaccine. I explained it. These are the things, and some of the other myths. And immunizations saves lives and keep people healthy. And the definition, I explained it just now. And this is the maybe I will start from this slide. Maybe you are seeing this slide first time. Yeah, I think because this is very essential to explain later slides. Okay, so I will explain once more. So when you are injecting a vaccine, these are the viral antigens. We are injecting with some adjuvants for getting a proper reaction. So this will be identified by cells called dendritic cells. These dendritic cells are also called antigen presenting cells. These antigen presenting cells will be the first cells to identify the antigen, and these cells will give the information to T cells. T cells will be like two type, CD4 T cells and CD8 T cells. These CD T cells also called T helper cells. T helper cells will activate a group of cells called B cells. These B cells are actually the main player. They will secrete the molecule called antibodies. This antibody can bind it to the specific pathogen, and this antigen they will differentiate into two types of cells: memory B cells and plasma cells. This antibody secreting B cells are called plasma cells, and some part of this memory B cells and plasma cells stay in the bone marrow for expecting later infection. And similarly, CD8 cells will differentiate into effector cells or killer cells, and some of the memory cells will stay in the bone marrow for later infection. So I am telling about the complications associated with the COVID-19. It is not just like a fever or flu. It will affect our multiple organs system, including heart, reproductive system, neurological system, or psychological system, immunity, our kidney, digestive system. So it have a high chance of multi-organ failure, and we don't know the long-term complications of this disease. So at any chance we should avoid the chance of this infection. And this is the two vaccines mostly used in USA, Pfizer biotech and Moderna vaccine. These both vaccines are based on mRNA. I told why we are using mRNA because mRNA synthesis are considerably easy manufacturing process. Protein synthesis is not that much easy, but because of the instability of this mRNA, we need a lipid nanoparticle coat for stability reasons, and this red color spike protein will be coded by this mRNA. So when we injecting this lipid nanoparticles vaccine inside a cell, and this cell machinery will use our protein synthesis. We have a robust protein synthesis machinery in our body is called ribosomes. These ribosomes will synthesis this antigens or viral antigens. So some of these antigen will express in the outside of the cell, and some when the cells dies, these proteins will be come out of the cell, and this will be identified by our antigen presenting cells, and this information will pass to helper T cells, and helper T cells will activate the B cells. They are the actual antibody producing cells, and they will secrete so much any number of antibodies. This can bind it to our coronavirus because this is having a similar spike protein, and they are specific to that spike protein. This can inhibit the virus entry into our cells, and same time I told this there's another T cell population. They're called the killer T cells, which can actually destroy the virus infected cells. Means this after entry of virus in our cells, they will multiply and they will be packed inside a cell. So that type of cells will be destroyed by these T killer cells. This is the mechanism of Moderna vaccine. And because of the stability issues, we cannot give this vaccine to all, like especially like in India type of country, because this we need a -80 or that type of refrigerator. We don't have that many number of refrigerator. So we the other option is DNA vaccine, but DNA vaccine is highly stable. So in this technology, the Oxford AstraZeneca used a technology called adenovirus mediated delivery. These adenovirus are non-replicating, non-pathogenic, and it is specific to chimpanzee and not to human. So we'll put that spike protein DNA into that adenovirus, and this adenovirus will be injecting to the cell. So this adenovirus are not harmful, so they will enter the cell, and this DNA will be released in the nucleus. They will be synthesized as mRNA. Then this mRNA can synthesize protein by our translating protein machinery. So the similar way, the spike protein will leads to the immunity. As I explained the previous slide, These are the two new generation vaccines mostly using throughout the world. And these are the some of the parameters, such as the boosters. Like this, all vaccine need 2 boosters for proper immunity. And these Sputnik and Oxford vaccines are viral vector vaccines, and these are most of them are like more than 90% effective. And because of this, I told the storage temperature, this we needs to keep these 2 vaccines at low temperature, but the other vaccines we can store it even regular fridge temperature. And this maybe the Covaxin and Covishield. These are the major vaccines we are using in India. Actually, the Serum Institute have a collaboration with the AstraZeneca Oxford vaccine. So they are also using the non-replicating viral vector adenovirus vaccine. And it can store in 2 to 80 degrees Celsius. And it requires 2 doses. And this is about the Covaxin. Actually, this is developed by, this is actually indigenous vaccine. This is actually used another methodology called inactivated virus. Actually, they inactivated the actual coronavirus. This methodology is not a new thing. It was used previously in the case of rabies and polio. We are still using that inactivated virus. And the initial paper shows that these both vaccines are highly safety. So there is no worry about to taking these 2 vaccines because their highly safety profile is very good. And then I'm telling why this safety is important. And this is approved by like a different step of clinical trials, mostly 3 or 4. The first clinical trial after animal studies will go for a 20 to 100 healthy volunteers clinical trial. And in this case, the major aim to check the safety of the vaccine, and we will check the how this vaccine is safe, or is have any serious side effect. If this is okay, then we will go for the second trial. In this case, we will check the short-term side effect and immune reactions. Actually, the first 2 will be, it's not actually looking the efficacy of the vaccine. We look for the safety. Actually, when we are giving this vaccine to a healthy people, the safety is the most concern, because they are not a diseased people. So any adverse effect will be a huge impact. So in the phase 3 clinical trial, we will go for thousands of volunteers. Actually, in that case, we will check safety and the effectiveness, whether this vaccine is protecting people compared to the unvaccinated people. If this third clinical trial is okay, then we will approve the vaccine for clinical use. And still, even after the approval also, it will not stop this monitoring. The manufacturing, our company are responsible to monitor in the long-term basis. They should report the regulatory authorities such as FDA in regular intervals about the vaccine risk and any adverse effects or the effectiveness. So this is like a continuous process. So these all rules are there to make the vaccine is safe. So we don't have to worry about the safety about our current vaccines. And maybe you think how much time will take to develop a vaccine. These are the some of the historical things. This year indicates that this is the first infectious agent identified time. For example, in the case of meningitis, like we identified that it is caused by Haemophilus influenza in 1889, but 1981 only we approved the vaccine. So it took around 100 years for making a vaccine. And in the case of polio also, it took 50 years for making a vaccine. So this is vaccine making process is not a easy process. But in the case of corona, it's in the whole vaccine history, we made a vaccine and we done 3 clinical trial within a calendar year. That means within months we made the vaccine and made available to clinical use. This is a very remarkable achievement of our medicine field. We should appreciate that. And maybe that vaccine effectiveness, I told this is 95%. What is that? value means. So like some people will think that the 90% means if I am getting a vaccine, I have a 90% chance that I won't get the disease, means I am getting 90% protection. It's not like that actually. If I am like if we are giving vaccine to 100 people, like out of 100, how many people will get disease? This is the sum of the like how many proportion will get disease compared to the unvaccinated people. So it's a proportion. Maybe the like 90% means like 90 people won't get disease, but still 10 people can get the disease. So that is the understanding. So this equation, like VE means vaccine effectiveness. This is like ARU minus ARV divided by ARU into 100. ARU means attack rate of disease in vaccinated people, means how many people are getting disease in vaccinated group minus how many people are getting means unvaccinated, U ARV vaccinated group. So if you divide it by this with the ARU in both side of the equation, we'll get ARU by ARU 1 and ARV by ARU is called relative risk, and we will get a new equation called vaccine effectiveness 1 minus R relative risk. So the vaccine effectiveness will always will be less than 1. So 90% means 0.9, 0.8 means 80%. This is the calculation. And for example, the basic calculation will be like that. Like we vaccinated 10 people, like 10 out of only 2 people got the disease. The unvaccinated population, 9 people got disease out of 10. So this is the like proportion 0.2 and 0.9. Then the vaccine effectiveness will be 78%. This is all about the vaccine effectiveness ratio. When you hear about vaccine effectiveness, this equation should be remembered. And you may heard about herd immunity. So what it means? So before I am telling this what is herd immunity, you see the color of the people. Red means diseased, blue susceptible, like orange vaccinated. Just assume that one diseased person with a corona is coming to a healthy population. So after some time, this person can give disease to all of the non-vaccinated population. But in another population, we have a vaccination, but not all people. Some of the people have vaccination. So and the same at the same time, one corona infected person came to that population, but still that person can give all susceptible people, not the vaccinated people, and some people won't get the disease. But if we can in the another case, we give like vaccine to maximum number of people. So here susceptible peoples are very less. If that infected person is coming to a population, the getting chance of disease to this unvaccinated people will be very less. That means even if you don't have a vaccine, your community have a vaccine, in number of vaccinated people, you will be get protected. This concept is called a herd immunity. This is what we want to achieve by vaccination. In the corona case, we don't know how much percentage we need to be vaccinated. Maybe it's 90% or 70%. People are telling different value, so it will take time. And I will tell about some history about vaccination, because we should know about the history to make some good decision for the future. And this person you might be heard like Edward Jenner. This is the person first. He's widely known as the father of vaccination. He vaccinated a kid in 1796. So actually this vaccination concept, I want to tell some background story like that. It is not, he is the not the first person conceived this idea. Actually, even in the early, even 2000 years ago also, India and China, they practiced a medicinal practice called variolation. In the case of smallpox, they will collect it pus from the infected person and inject it to a healthy people. And the because it's very highly risky, because this virus will may leads to death in so many people, but the recovered people will be immune to smallpox. And this was this was went to even Arabian region and all, even Turkey and all. So one wife of one ambassador to Turkey by like British person lady, she understood this concept in early 70s, and she took this idea to British, and she started doing this variolation procedure. but people are not very satisfied with that because high risk. But this person knew this idea, and he also observed that even some of the people working with the cows also having getting smallpox, and they are immune to disease. Then he thought that that disease can virus can or that disease can give some protection, so he collected this thing from virus from a cow, and he started giving. So instead of the actual virus, he giving a less harmful virus as a vaccine. So that is called that is the start of vaccinology. Even vaccine name also, vaxa means cow, you should understand that. And this person is Louis Pasteur. He is known for anthrax and rabies vaccine, and you can see some of the movies about him. He's a very remarkable person in medicinal field. And maybe you saw this iron lungs or artificial machine ventilators. So in the 1930s, in the US, having a high pandemic of polio, and this is a neuromuscular disease, and these people cannot breathe themselves, and they need a constant support of these artificial ventilators for life. And this is the some of this very recently 1930s, this was the condition. And this is the person, Jonas Salk, who first one of the first person developed this polio virus. Actually, in 1953, what he did is that he cultured this virus in monkey kidney cells, and he isolated that virus and inactivated by formalin. And then he started a clinical trial in US and Canadian regions in 1.5 million kids. And in 1960, within 6 years, he reduced the infectivity or infection into 95% in US. So he's a remarkable person in polio virus field. And another person, this person is not known too much. His name is Maurice Hillman. You might be hear about vaccines such as measles, mumps, MMR vaccine, hepatitis vaccine. Almost 40 vaccines developed by him, and he's credited with saving more lives than any other medical scientist in the 20th century. He's a genuine hero, yet most people wouldn't recognize his name. And we have evidence that why these vaccines are important, because in the 100 years ago, these are the top 10 cause of death out of 1 lakh people. We can see most of them are like infectious disease which can be prevented by vaccine. When it comes to 2010, these vaccine preventable diseases are very reduced. Now the cancer and heart disease are the most prominent cause of disease. So vaccine have an important role to change this trend from infectious to lifestyle disorder. And the total number of deaths in 100,000 people also reduced very considerably from 100 in last 100 years. And regarding the life expectancy, 100 years ago, the most of the countries, the life expectancy was less than 50 years of age. We can see the India. And when it comes to 2018, we can see in the some of the countries like Australia and some European regions, this life expectancy went even greater than 85. These vaccination also have a great role in this trend. And by the way, in the case 100 years ago, our life expectancy was 24-year-old. That means it is very less than more than like our age. Like we used to tell that we had a good past or something, like people are more healthy in the past days. And it's not like that. Child mortalities was very high in those days. Even the independence time also, our life expectancy was less than 40, and recently only it is went up to above 60 because of a lot of vaccination programs happened after the independence in India by our government. And why I am telling this vaccination is important, some of the recent data came from US. This indicates that this colored bars indicates that these are the vaccine preventable diseases in children and mortality. And that means in the 1990, around 5.5 million child deaths are due to vaccine preventable disease such as tetanus, pertussis, measles, etc. But when it comes to 2070, this is reduced to 1.8 million. So vaccine is working very nicely, which reduces our child mortality, which increases the life expectancy rate. And these are the some of the vaccine approved in USA, and this is the percentage of cases reduction, and this is the percentage of death. So if you see this, almost all are like 100%. That means if you have a vaccine, you is like you won't get the disease and you won't die because of that disease. That much sure is there, like the most of the vaccines now. And like this graph gives a very hope for us for regarding corona infection. Some of the diseases, like this is the disease number of persons infected with disease by year. For in the case of diphtheria, in the 1940, the vaccine first vaccine was introduced. We can see a clear decline of number of people infected after this vaccine introduction. And this is happened to polio, meningococcus, Haemophilus influenzae, measles, pertussis. So this having a same trend. This all vaccine helped us to eradicate this disease. This shows that the current corona vaccine also will be a successful strategy. And the one example I will show, a polio virus in 1950s. The red color indicates that the infection endemic to polio. The all means the whole world is infected with the polio. When it comes to 2070, only some part of Africa, our neighbors such as Pakistan and Afghanistan, they only have polio. In fact, polio endemic countries. Other all countries are now polio free. So this is also a very successful story of vaccination, polio. And I will say that India, regarding vaccination, we are proud to say that we have a rich history of vaccination. Even the first vaccine research institute established in Himachal Pradesh in 1904, around 100 years ago, and first BCG vaccine laboratory set up in Madras in 1948. And India developed the first indigenous oral polio vaccine in 1970, and first ever recombinant DNA hepatitis B vaccine developed also in India in 1997. So we had a good history. I'm just telling some of the example only. And regarding the disease, from 1980 to 2010, we reduced the number of cases of disease such as diphtheria, total tetanus, polio, measles, pertussis, etc. Through vaccination. And I want to say that we have a n number of good vaccine production companies in our country, and they are very reputed, and this is running for a long time. And for example, you may heard about Serum Institute of India. It was established in 1966, so they have more than 60 or 50 years of experience in vaccine. And Bharat Biotech was established in 1996. They are making vaccine for COVID vaccine for us now. And they are not only making vaccine for us, they are making vaccine for other lower income countries, because in a affordable price. That is also they're doing a good job for the entire humanity. So we should appreciate that. And the another two success stories regarding the polio smallpox eradication. We won't see such type of child now. Actually, the smallpox vaccination in India started even just 6 years after, when Edward Jenner found this vaccination method through British government. It came in India. There is a small stories actually there. So actually, this vaccine came through like Mumbai port, just as in like cow thymus extract. So when they started this vaccination, like some of the conservative people, like they are telling this using this cow extract will be against their religious belief, and some protests will happen that time. That time also is vaccine hesitancy, even say that, prevalent in our country. And that's some of the historical fun fact. And 1962, National Smallpox Eradication Program launched. And 1975, within like 15 to 13 years, last case was reported in India. And actually, it is a note from He's not an Indian. He came from Bangladesh. This is a report in Assam. And 1977, India declared smallpox free. And 1980, World Health Assembly declared the world free from smallpox. And this is the one disease we eradicated from the entire planet. So this is a highly successful story of a vaccine. And another one is the polio. We won't see this type of kid in our generation, at least, because this vaccination program. And 1988, World Health Assembly set a target of polio eradication by 2000. And especially Tamil Nadu and Kerala state took initiative in conducting special drive to polio, even after that only the national immunization day started in 1995. And in 2011, this was also very successful. Last case was reported from West Bengal in 2011, and by 2012, WHO removes India from the list of polio endemic countries, and this is a success story. But the another thing, if you see, because of diversity and socio-economic difference and access to healthcare facilities, we don't have a equal amount of vaccination in all part of the India. So the next goal of our India should be to vaccinate these all people equally, and some part even below 40% of the kids are getting vaccinated. And the current COVID also, it's a recent news came in the Time magazine, Time Economist magazine, because when we can give maximum number of vaccine in each countries, maybe in the US and all, the late 2021 they can give vaccine to most of the people. But in the case of India, we have to wait the late 2022. This is the predictable date we can give a maximum coverage in the population. So, but in the case of Africa and other regions and our neighboring countries like Pakistan and all, even they will not finish by even 2023. So the thing is that if you don't have a vaccine, the entire population, this virus will be here for a long time. These are the some of the predictions. So as this is my end of the presentation, like Nelson Mandela told, life or death for a young child too often depends on whether he or she is born in a country where vaccines are available or not. So vaccines are such an important thing, and access. Okay, so I'm ending the presentation here. You can ask the questions in Malayalam or English. I will try to answer. Okay. Okay, the first question came, what percentage of population needs to get vaccinated to have herd immunity to COVID-19? Actually, it is predicted that when 70%, but even 70% also, if you think Indian population, it is that number is like more than around 100 crore. We need to have that many number of vaccine. That is the problem.
Okay, I have a question. Immunity after affecting with COVID-19 or protection from COVID-19 vaccines, which lasts longer?
Yeah, it's actually a nice question, but the first answer is that nobody knows now, because it's like our vaccine came maybe some 6 month or 2 month. So we need to check the antibody level after like 6 months and all. And those who are infected with the COVID also, we have to check after 6 months or 1 year. And in the case of our previous experience, our previous study shows that it can go to either way. The some of the vaccine, so vaccine will work better than the natural immunity. But in the some of the cases, it can go the other direction. The natural immunity will give more protection. So it can go actually either way.
Oh,
Yeah.
okay. Thank you. Thank you, Dr. Aneesh, for such an interesting and thought-provoking session.
Yeah.
Especially your talk was appropriate at this point of time for COVID-19 pandemic spread.
Okay.
It was very pleasure to listen to your talk. Thank you once again.
Yeah. Okay. Yeah. Thank you for inviting me. Yeah. Some technical issues. Yeah. It's like.
I apologize for all the
Yeah.
technical problems.
Yeah.
So,
Like. Okay.
so here comes to an end to our sessions. Hopefully, wishing that all of you have something to take home. I don't want to elaborate and put you into boredom. Without much delay, it is my privilege to propose vote of thanks on this occasion. First and foremost, on behalf of St. Thomas College, a big thank you to Dr. Tapas and Dr. Aneesh for considering our invitation without a second thought. We wish that someday we can meet you directly to take offline sessions in our college, but not online. I must mention our deep sense of appreciation for sparing your valuable time with our students and other participants and sharing your findings and opinions with us. I think already it's late night for them. Maybe 1 a.m. or 2 a.m., right?
It's 2 a.m.
Hope you get some sleep. It must be late over there. I would like to thank our Principal, Dr. Joy KL, for his wholehearted support. And my sincere thanks to Vice Principals Reverend Dr. Martin Columbret, Dr. Joby Thomas K, Dr. Sister Alfonsa Matthew, IQAC Coordinator Reverend Dr. Anil George K, Dean of Sciences Dr. Brito Joseph, and College with Potential for Excellence team for their rock-solid support system and inspired us to do our best. My heartfelt thanks to Dr. Vimala Jose, Head Department of Botany, and all other faculties of Department of Botany for taking such an initiative for conducting such a webinar. Thank you to all the participants for their esteemed presence and wishing this webinar was informative. Once again, thank you very much. Thank you.